just continue whenever you're ready.
Okay, yeah. So good morning, everyone. As mentioned before, my name is Hammond, and I am a young researcher at the division of. Oh, the screen stopped sharing. I don't know what's going on.
Maybe try to do it again. Just try to reshare the screen.
Yeah. I have my other computer open, so maybe I'll share from here. Makes more sense. Sorry, guys. I don't know what's going on. So share screen. I don't know. Doesn't give me the option. Everything is in Czech. Sorry, I'll try again. Share. Yeah, can you see it?
Yeah, something is going on. Yeah.
Perfect. So yeah, so as I was saying, I am a researcher at the division of plant physiology and cryobiology at the Crop Research Institute in Prague. And the research I will be presenting today is the development of a PVS2 plant vitrification method for cryopreservation of yacon, which is a plant. Its Latin name is Smallanthus sonchifolius. But yeah, so it's not working. I am pressing, but it doesn't go. I don't know what's happening. No. Sorry, I don't know what's happening. It doesn't work.
We can see your first slide, and probably if you need more time, we can come back to you after another report so you can try to fix it. How does it sound? Or you can just present without the slides, so I don't know, to share one by one each of them.
I can't hear anything. I don't know if you can hear me. Oh, I don't know what's happening.
We can hear you, but we can see on the first slide of your presentation.
I will stop sharing. Maybe. And sorry, I don't know what's happening.
You can skip this first slide with the video.
Okay, it seems to.
Because video requires a lot of good internet connection, and if you just skip the video, we can probably move on.
Yeah, everything is crashing. I don't know what's going on. So, yeah. And I can share the presentation later. I don't know what's happening. Or I can share the link to the presentation, maybe.
Yes, you can just share the link for presentation afterwards, so don't worry, just can explain to us what is the topic of your interest, your results, and then we can take a look.
Oh, yeah.
It happens, so don't be stressed.
Yeah, so basically my research is about cryopreserving this Andean tuber called yacon. The reason why I decided to work on this tuber is, well, it was my PhD topic, and I am from Latin America, so it made a lot of sense. And this Andean tuber is the third most important or economically important crop in South America because of its economic value, and also because of the fact that it's one of the only tuberous crop species, or tuber and root crop, that can be eaten raw without being cooked. And from a scientific important point of view, it is one of the largest natural source of inulin type oligosaccharide, which is a great replacement sugar for diabetic people, as everyone knows. So yeah, so the plant basically produces two types of tubers, or storage tuber and the rhizome, which is the propagating material, but it is a sterile plant, so it's vegetatively propagated. So up until I did this research, there was no long-term conservation scheme or mechanism or approach to conserve the species. It was mainly being conserved under field conditions or by farmers in the Andean region. And in the International Potato Center, they developed a slow growth in vitro conservation method, but it doesn't guarantee, or it was not long term. So we decided to test cryopreservation. And to do so, we decided to use, as at our institute, in our team, we focus on the PVS tree plant vitrification method for conservation. This method, basically, we did a 50 to 50 or 50% glycerol, 50% sucrose vitrification method or cryoprotectant solution. And we tested different time of dehydration. So we needed to dehydrate the cell as well. Thankfully, everyone here knows I usually have to explain it, that you have to reduce the water in the cell to avoid crystallization or cell crystals and damage by frost. So I tested several different times of dehydration from 15 minutes to 120 minutes. So with 15-minute intervals. We found that the preliminary results showed that 60-minute treatment time duration was the best to reduce the water content. So we then tested this protocol on 5 different genotypes of yacon originating from Peru, Bolivia, and Ecuador. To do the preliminary testing, we tested, or we used the Ecuadorian accession. We did the different dehydration time, or we tested the different dehydration time. We found that 60 minutes was the best. And then we had to go on to the recovery phase and develop appropriate recovery media. So we tested 2 different approaches. One was Murashik and Skog media, which is a basal culture media as baseline for recovery. And then we also tested this media with the addition of benzylaminopurin to induce shooting of shoots after cryopreservation. Our results in terms of the recovery, in the results we found that the shoot tips were able to recover on both media. However, the benzylaminopurin media, or benzyladenine media, sorry, there was a high level of hyperhydrosity and callus formation because when we associated this with the addition of benzylaminopurin. So the overall optimum media was, recovery media was MS, or Murashik and Skog media without the addition of benzylaminopurin. So now that we know that the 60-minute treatment time duration was the optimal dehydration rate or the dehydration treatment, and MS media without benzylaminopurin was the best recovery media, we applied this to the other accessions of yacon, so 2 from Bolivia and 2 from Peru. Overall, our results found that it took about, from cryopreservation to recovery, it took 12 weeks to obtain fully regenerated plants, meaning with well-developed roots, at least 2 to 3 well-developed roots, and at least 2 to 3 well-developed pairs of leaves. We found that the recovery rate range, or the survival rate, which you can determine within the first 2 or 3 weeks after cryopreservation, ranged from 80 to 94% within the different genotypes. And up until the 12 weeks of recovery, we found that the fully grown normal plants with the criteria that I mentioned before, the well-developed roots and leaves, we had around 62, well, between 62 and 75% recovery rate. We consider this experiment or this protocol to be highly successful as this, at least in our lab, I guess it differs in different gene banks. In our lab, the minimum acceptable survival or regrowth rate is 40%. So having 60% and more is considered as quite successful. In conclusion, we found that using the Ecuadorian genotype, we proved the feasibility of PVS3 as a long-term storage method for yacon. We found that overall a 60-minute treatment time duration of PVS3 and MS media without addition of benzylaminopurin or without the addition of any hormone was optimal in an optimal protocol to be used and be applied in different accessions of yacon. Once applied, we also proved the feasibility of this protocol to be use, wider range of genotypes or yacon genotypes originating from different areas within the Andean region. So that's it. I tried to summarize as quick as I could. I am so sorry about the presentation. I will share the PowerPoint with you guys in a little bit. Yeah, once I figure this out. So,
Thank you so much. I can assure you that even without the slides, it was a great talk, like really one. And I want to congratulate you on such wonderful results because it's a true success. With such survival rate, it's like really impressive. And we have a lot of questions for you, actually. So the first question from Brian Grout. And do you have plants growing successfully towards the maturity? And when will you know if they can complete their life cycle?
I tried to summarize. So we did a full experiment. I will not go too much in detail. So what we did was we cryopreserved. I had it in the presentation. I just skipped a lot of stuff. But I have collected the samples from South America. Actually, this is my PhD studies at the Czech University of Life Sciences. And there, in their field collection, they have the largest European collection of yacons. So basically, I put the collection from South America in this field collection as well to have more plant material so I can collect apical shoots to establish in vitro. I established the plants in vitro, propagated, have enough material for cryopreservation, cryopreserved. Then I also transferred the plants to ex vitro condition and did the whole life cycle as well, because I also did chemical analysis, growth, the growth of the plant and stuff like that, which I did with as a part of a MSc students research. Because, well, once we reached the cryopreservation, I didn't need to do the genetic analysis, chemical analysis, but we wanted to test this theory. As most of you know, or all of us know, in cryopreservation, we consider that the metabolic processes are at a standstill and there should not be any genetic variation or stuff like that. But I did do this, like take the plants to ex vitro and did the full recovery as a part of this additional research to test this theory. So in conclusion, yes, we did the whole cycle again. Of course, under these conditions here, because this is an actual perennial crop. So under the conditions here, because the plant does not withstand frost in field condition, what we did was we planted them in the field after the last frost in the soil, and we harvested them in about May, and we harvested them before the first frost in the soil. So somewhere in October, early November. So yeah.
Thank you so much for this explanation. We have plenty of nice comments in the chat about your work, and also I can see the technical probably questions about the protocol, about the content of the solutions that you use for vitrification, and also about type of containers. But I guess you can just share with us the link to a presentation, and in this case we will know all the information. So please don't forget to do it and add your presentation to the comments. And now we probably will move on to another speaker because it's a time, and well, it's not